Let's go down to where we were. We were here. Okay. Everyone see this, correct?
Yeah.
Yes.
Good. Okay. You got to talk. Remember, I can't see you. You got to talk. All right. And at any time you have trouble, stop me. All right. Let's talk about platelets. Platelets come from megakaryocytes. These megakaryocytes, which are cells, split off into 2,000 to 3,000 fragments to create platelets. So that platelets are not cells, they are fragments of cells. Their sole function is clotting. That's their sole function, to help your blood clot. We'll talk about clotting in more detail, but platelets is only one item that helps you clot. Under the influence of the hormone thrombopoietin, stem cells differentiate into platelets. What they differentiate into a megakaryoblast. Those megakaryoblast become megakaryocytes. Those megakaryocytes fragment into platelets. Here's another hormone you have to know. We talked about the hormone erythropoietin. What does erythropoietin do? It stimulates the stem cells to produce erythroblasts. Erythroblasts eventually produce erythrocytes, and those erythrocytes are known as red blood cells. Okay, so you have erythropoietin and you have thrombopoietin, two blood hormones that you have to know. The lifespan of a platelet is between 5 and 9 days. I use the term 7, a week. Platelets survive 7 days on the average. Now, why is that important? Well, I'll tell you why it's important. You're young, you go out and you play a game of basketball, you play a game of tennis, you play a game of baseball, and you tear your medial collateral ligament of your knee. You need surgery, but you're taking aspirin. Okay? The orthopedic surgeon will tell you aspirin destroys platelet function. I cannot operate on your knee to save your medial collateral ligament until you stop the aspirin. What's the lifespan of a platelet? 7 days. Therefore, I cannot operate on you for 7 days. Everybody understand that?
Yes.
Good. Thank you. Got to talk to me. All right? Okay.
I have a question.
I'm sorry?
I have a question.
Go.
So when you donate blood, then like sometimes they only take like you can they have options where they only take the platelets, but you still can't donate blood for 120 days. Is there any reasoning for that?
Correct.
But if the platelets replenish itself every 5 to 9 days, then why wouldn't you be able to donate platelets like every week or something like that?
Because they're taking your whole blood and they're spinning it down. So they're just using the platelets, but they can't just draw the platelets out of your body. They draw your blood, correct?
Yeah.
So they also, and trust me, they're going to use your blood. They're going to lose your plasma. They're going to use your red cells. I guarantee it. I guarantee it. Even though you're just donating platelets, but you can't just donate platelets. You have to donate your entire blood, and then they spin it down and take the platelets off. Does that make sense?
Yeah, Thank you.
And let me ask the class this question now that you brought that up, which is a very good question. Where are the platelets in the spun blood? Are they in the plasma? Are they in the red cells? Or are they already in the buffy coat?
They're in the formed elements, right?
No, they're not.
Aren't they in the Buffy coat?
Yes, they're in the buffy coat. Their platelets are in the buffy coat. What else is in the buffy coat besides platelets? There's one more thing in the buffy coat.
White blood cells.
Yes, white blood cells. Okay? Every time you donate blood, you're donating plasma, you're donating platelets, you're donating white blood cells, and you're donating red cells. They use every bit of it. Every bit of it. Everybody understand that?
Yes.
Got it. Okay, here we go. A review. This is a review. We talked about the white cells and the red cells. Obviously, there are more red cells in your blood than any other cell. Okay? And the function of a red cell is to carry oxygen. And it is hemoglobin that carries the oxygen. What is the mineral basis of hemoglobin?
Iron.
Thank you. Iron. All right. I'm asking you these questions because you're going to see them again on exam number 1. I'm trying to give you 85% of your exam. So listen up, okay? Or go to YouTube later. All right. White cells. You have neutrophils, eosinophils, monocytes, lymphocytes, and basophils. Remember, never let monkeys eat bananas, from the most common to the least common. Okay, here it is. 60% of all white cells are neutrophils. And you can certainly review this. We've talked about it already. Okay, there they are. We're going to talk a lot more about lymphocytes when we get into chapter 22. That's the basis of your immune system. All right? So that's important. And we just talked about platelets, and platelets are fragments of cells. Very important. Okay? Bone marrow transplants. This is something I'm not going to really talk about. You don't need to know about bone marrow transplants. We'll talk about other things more important than bone marrow transplants. But what we need to talk about is how to stop bleeding. We touched on it already. We touched on platelets. Platelets will stop you from bleeding, but it won't stop 100%. Here is the process of what we call hemostasis, the sequence of responses that stop bleeding. The process involves vascular spasm. When you're cut, the arteries and veins that are cut go into spasm to try and slow down blood flow to that area that has been injured. That's known as vascular spasm. The second thing that happens is that platelets come to the area, and that's called platelet plug formation. And the third process to stop you from bleeding is called coagulation, otherwise known as blood clotting. So the three mechanisms of hemostasis, stopping bleeding, are vascular spasm, platelet plug formation, and blood clotting, known as coagulation. Platelet plug formation. Platelets come to the site of bleeding. They adhere to the walls of the vessel, the walls of the tissue. They liberate hormones that stop bleeding, and those hormones and neurotransmitters are right there. ADP, adenosine diphosphate, serotonin, and thromboxane A2. This is liberated by the platelets. And then you get more platelets, and there's now a platelet plug. And that's called platelet plug formation. Only 1/3 of hemostasis. First is vascular spasm, second is platelet plug formation. When you actually get a blood clot formation, it is actually caused by fibrin. Remember, in the plasma protein section of the plasma, there are 3 proteins: albumin, globulin, and fibrinogen. Well, now we talk about fibrinogen. Fibrinogen is converted into fibrin. Those strands will give you a blood clot. All right? So we need to talk about the formation of that blood clot. It depends on several clotting factors. It's called a cascade. This clotting is activated in 2 ways. It's either activated by an outside pathway, outside of the blood, or an intrinsic pathway, which is inside the blood. So you have a tissue pathway and a blood pathway. Both pathways lead to the formation of an enzyme called prothrombinase. And it's from there that you now get a common pathway. I will show you that slide. Here it is. Extrinsic pathway, tissue trauma. You get a cut in your arm. The tissue of your arm sends a factor into the bloodstream. It's called a tissue factor. Along with calcium, which is the most abundant mineral in your body, it activates factor 10. Factor 10, along with calcium, will stimulate the formation of prothrombinase. That's the intrinsic pathway. The intrinsic pathway is when the vessels of your blood have been compromised. You now have platelets coming to the scene. Along with calcium, the most abundant mineral in your body, and platelets, factor 10 is activated. In addition to calcium, you stimulate the production of prothrombinase. So you have the extrinsic pathway, the intrinsic pathway to give you prothrombinase. This is now the beginning of the common pathway. The common pathway. Next Tuesday, I will give you a better lecture on blood clotting. Okay? So you won't be so confused. But this is the common pathway. Prothrombinase produces thrombin. Thrombin is the enzyme that will stimulate fibrinogen into fibrin, causing you to have a blood clot. Now the blood clot is formed. Thrombin, fibrinogen to fibrin. Okay? We'll talk a lot more about this on Tuesday because I think this is a very important thing to understand. And it just doesn't hit me that it tells you exactly what you should know. All right?
So extrinsic pathways are, I'm like, what kind of like injury would be like extrinsic pathway for like your body to heal, and what kind of injury would be like an intrinsic pathway to heal it?
Well, you've got to remember something. What if I cut your arm? Okay, say I cut your arm with a very sharp knife, and you needed deep enough injury to need stitches. Okay, what does the knife go through? The knife just doesn't go through your vessels; it goes through your tissue. So that's what we're talking about. That's the extrinsic pathway. The extrinsic pathway is the tissue of your arm. The intrinsic pathway are the veins and arteries of your arm. Does that make sense?
Okay, I get it now.
You understand?
Yeah, thank you.
Oh, okay. All right. All right. These are the total factors that are in clotting. Really, these factors are made in the liver. Okay, these factors are made in the liver for the most part. For the most part, what do you really have to remember? Well, you're not going to remember 13 factors. I can't even remember 13 factors. Okay, but you got to remember fibrinogen because that's the plasma protein that is involved in clotting. You also have to remember calcium. Without calcium, your blood will never clot. Never. Okay? Calcium is the most abundant mineral in your body. I think I've said that 10 times already. If I say something more than 3 times, you'll see it on your exam. All right? The only real other factor that you have to remember is factor 8. Factor 8 is the anti-hemolytic factor. It is the problem in a disease called hemophilia A. And you will have a student lecture on February the 11th, on hemophilia A. So you should know fibrinogen, calcium, and factor 8. Factor 8. There are other things in here you don't have to remember. That's not important. That's not important. All right? Got it? Somebody say yes.
Got it.
Good. Good. All right. Calcium is the mineral involved in clotting. Once the clot forms, it tightens. Without vitamin K, you cannot clot. So if you have a deficiency in vitamin K, your clot formation will not be good and you will continue to bleed. You will continue to bleed. So calcium is the mineral involved in clotting, and vitamin K is the vitamin involved in clotting. All right? Very, very important. Very, very important. I will talk about plasmin next Tuesday because the reason plasmin is important is because what if you want to get rid of a clot? Well, this enzyme gets rid of it. Gets rid of it. It's known as a fibrolytic enzyme, plasmin. Thrombin makes the clot, plasmin gets rid of it. And we'll talk more about it on Tuesday because we use this in medicine to get rid of a clot. All right, let's talk about blood groups. There's a lot of blood groups around. Okay? Basically, blood groups are based on the presence or absence of agglutinins or glycoprotein and glycolipid antigens on the surface of red blood cells. There are more than 24 blood groups and more than 100 antigens. We discover them every single year. Okay? what I want you to know about blood groups is I want you to know the ABO system. You're either type A, type B, type AB, or type O. And that is very important to understand. Okay? The other antigen I want you to know is the rhesus antigen. You know it is RH. That abbreviation comes from the rhesus monkey, where it was first discovered, RH factor. The majority of the world is RH positive. That means you have the antigen. And here it says 85% of humans. Well, 85% of Americans. When you come into the Asian group, 100% of them have it. Okay? So the majority of the world is RH positive. The minority of the world is RH negative.
So what would happen?
Go ahead.
What would happen if you didn't have the RH antigen?
I'm going to tell you in a minute.
Okay.
I'm going to tell you in a minute. If I don't, remind me, but I'm going to tell you in a minute, okay? You know, before we leave this, it's really too bad I don't have a whiteboard here. I have to figure out how to do a whiteboard, but I think I'll figure it out. What's important to understand is that when you say you are A, let's say my blood, and I can tell you right now, I know my blood type. My blood type is A. You have to remember that half of my genes come from my mother and half of my genes come from my father. Okay, so am I dominant A or am I recessive A? Am I AA, which is dominant, or am I AO, which is recessive? Does everybody understand that before we get into the nitty gritty here?
Yes.
Professor,
Dominant, go ahead.
then what are the like O positive, O negative, like what are the positive and the negatives of the blood types?
I'm going to talk about that. I don't want to confuse you with that yet. I'm trying to make you understand that not only are you A, you are either AO, which is recessive, or AA, which is dominant. Okay, got it?
Yep, thank you.
Let's go into B. You can either be recessive, BO, or dominant, BB. Okay? If you have an AB blood type, that's your blood type. AB is recessive and AB is dominant. Boom. If you're O, that's all you have. OO. Okay, so you're either AO or AA, BO or BB, AB or O. Everybody understand that?
Yes.
Yes.
How many of you don't know what a Punnett square is? Okay, that's genetics. That's what we're talking about here, genetics. We're talking about the Jerry Springer show, where someone comes on and says, I'm not the father of that baby. Well, guess what? This is why we take your blood. We take your blood and we can see if you are actually the father of that baby. So long as I know the blood type of the mother and the blood type of the father and the blood type of the baby, I can tell you who the father is or isn't just by doing that. Does that make sense?
Yes.
Good. Thank you. Remember, you got to talk. All right. So that's the ABO system. Now we got to talk about the RH system. Again, the majority of the world is RH positive. The minority of the world is RH negative. And we'll talk a lot about that in a few minutes. Here's what I was trying to tell you. Look at the Korean, Japanese, Chinese, and Native Americans. 100% of them are Rh positive. That means they have the antigen. Whereas Europeans and Africans are not 100%. So that's important. You don't have to know the percentage of OA, B, and AB. That's not important. Know that the majority of the world is Rh positive. That's the red cell. I want you to understand that is the red cell. Remember, when you draw blood, you spin it down and you spin it down into plasma, buffy coat, and formed red cells. So the plasma also contains antibodies that will react with the A and B antigens. An individual will not have agglutinins against his or her blood type. So here's what we want to show you about the blood cell and the plasma. This is a very important slide. Look at the blood types. In the red cell, type A. Type A has the A antigen, but in their plasma has anti-B antibody. Type B on the red cell has the B antigen, and in the plasma has the anti-A antibody. Type AB has both A and B antigens and no antibodies in the plasma. Type O has neither A or B antigens, but in the plasma has both A and B antibodies. By looking at this slide, I want you to tell me what blood type is the universal donor. I can give this blood to anybody, and they will accept it with no problem.
Type O.
Isn't it type O?
Type O, that is correct, type O. Because all I'm giving you is red cells. I'm not giving you plasma, I'm giving you red cells. So type O is known as the universal donor because it neither has A or B antigen on the red cell. Okay? Which blood type is the universal recipient? I can give this type blood, I can give anything. I can give them A, B, or O. Which blood type is the universal recipient?
AB.
AB, because it has on the red cell, it's got both A and B antigens. So what? So what? Okay? It doesn't matter. I can give, you can take everybody's blood. You can take O, you can take B, and you can take A because I already have the antigen. I already have it. Everybody understand that?
So if someone with type A gives someone with type B blood, what would happen?
They will take a die. Renal disease, blood clotting, fever, hemolysis, which is the breakdown of all red cells. It's extremely dangerous, and it is an obvious $1 million lawsuit. And you can't defend it. You cannot defend giving someone the wrong blood. You cannot defend it ever.
So you can't, like, say if someone with type A got type B, you can't survive that.
No, listen, but say you come into my emergency room and you've been shot in the chest, and your blood pressure is 40 over 0. Guess what? You need blood. You're dying, right? You're bleeding to death. Your heart's got a hole in it. Your aorta's got a hole in it. What blood am I going to give you immediately? I'm going to call the lab and I'm going to say, listen, I need 10 units of
Type O.
type O.
Typo.
Type O. All right?
Gotcha.
The
Thank you.
universal donor. I don't have to worry what your blood type is. I don't care what your blood type is. I need to save your life. Make sense?
Yes, thank you so much.
My pleasure. All right. Let's not forget about the rhesus factor, the RH factor. Remember, 85% to 95% of Americans, European or African, are RH positive. So basically, if you gave them O negative blood, you're a winner. Nothing's going to happen. But if you don't have any O negative blood and all you have is RH positive blood, O positive blood, you're taking a chance, but the chance is very minimal. Okay? So the universal donor basically is O negative blood because it's got no rhesus factor. But when I ask you the question of the universal donor on the exam, the answer is O. That's the answer. Okay, everybody got it?
Yeah.
Good. Okay, I don't even know what this means. This is ridiculous. You can see the interactions. I don't want to have any interactions. Trust me. I think I told you everything that I needed to tell you about blood types. Okay? You're not going to be typing anybody's blood, so don't worry about typing anyone's blood. We used to do this years ago, but now with all the violations of using human blood in a laboratory exercise, we can't do that anymore. Okay, but what I need to talk to you about is this disease. This is known as hemolytic disease of the newborn. When you are pregnant, sometimes, actually most of the time, either at birth or before birth, small amounts of fetal blood leak into the maternal circulation. Now, if the baby is Rh positive, which 85% of people are, and the mother is Rh negative, she will develop antibodies to the Rh factor, and the baby will be delivered with no trouble. However, during her next pregnancy, if she again has an Rh positive baby, when she transfers antibodies to the fetus, a normal occurrence, they transfer anti-Rh antibodies. It will attack the bloodstream and the blood cells of the second baby, will cause clotting and hemolysis. Hemolysis means the breakdown of red cells. If the baby survives, which is very rare, if the baby survives, it will be severely compromised. It will not be able to use its nervous system. It might even need amputations. This is known as hemolytic disease of the newborn. Here it is. The first pregnancy in an Rh negative mother who has an Rh positive fetus, the circulations entwine each other, and the mother now has anti-Rh antibodies. The baby here is fine. It's delivered. But in the second pregnancy, because of the anti-Rh antibodies, they will attack the red cells of the fetus and usually destroy the fetus and kill the fetus. And if not, it's a severely injured fetus when born. Does everybody understand that?
Yeah.
Yes.
To avoid that problem, we give all Rh negative mothers a protein called RhoGAM, RHOGAM, to prevent them from developing anti-Rh antibodies towards the second baby. So that's why they take your blood when you're pregnant, and you have to know if you Rh negative or not. If you're Rh positive, you're fine. If you're Rh negative, your gynecologist will give you a shot of RhoGAM, a protein to prevent you from developing antibodies to the Rh factor. Okay? There are other diseases. You're going to tell me about other diseases in your student presentations, and one of them is sickle cell disease. I am not going to talk about sickle cell disease because one of your classmates will talk to us about sickle cell disease. Okay? And I think that's it. It is. Good. Any questions at all? Let me stop the recording.